Hello everyone, hope you all are doing well. So today we are going to discuss the next question of our agriculture optional answer writing practice series, and the question for today is, how is detailed record of relationship between the selected plants and their progenies maintained in pedigree selection method, and discuss the procedure for pedigree selection method with the help of a simplified schematic diagram. So this is the question, and this question was asked in this year's agriculture optional paper, that is agriculture optional paper of UPSC Civil Services Examination 2025. So let's see how we can approach the answer. So in introduction part, obviously we can define pedigree selection, what pedigree selection is, and in the body you can divide the body in the two parts. In the first part you can mention how detailed record is maintained in pedigree selection, and in the second part we have to discuss the procedure of pedigree selection method using a map or using a diagram. So let's start. So what is pedigree selection? So pedigree selection is a method of plant breeding. Pedigree selection is a method of plant breeding. Okay, method of plant breeding, generally in self-pollinated crops. Generally in self-pollinated crops, in which the breeder keeps a detailed record of ancestry. Keeps a detailed record of ancestry, or you can say pedigree, detailed record of pedigree of the selected plant and their progenies, of selected plants and their progenies in each generation to identify superior lines over successive generation, to identify superior lines over successive generations, over successive generation. So what is pedigree selection? So it is a method of plant breeding generally done in self-pollinated crops, in which the breeder keeps a detailed record of ancestry. Here the breeder is keeping a detailed record of pedigree or detailed record of ancestry of selected plants and their progenies in each generation to identify superior lines over successive generation, so that through this record of pedigree we can know the superior lines in the successive generations. We can identify the superior lines in the successive generations. So this is what pedigree selection is, and this is how you can define pedigree selection. Now let's move to the first part of the question, that is how this detailed record of relationship between selected plants and their progeny is maintained. So how, or you can say maintenance of pedigree records. Maintenance of pedigree records. How this is done? So a detailed record of relationship is generally maintained in a pedigree record book. There is a pedigree record book where the detailed record of relationship between pedigree and their progenies, okay, between the selected plants and their progenies is maintained in this book only, pedigree record book. If you see the pedigree record book, you will find that each selected plant is given a unique identification code or number. Each selected plant is given unique ESP, that is each selected plant is given unique, okay, is given unique identification code or number. Unique identification code or number. For example, you can see that they will give like this: P1 1, plant 1 2, P1 3. That is in F2 generation. That is P1. Suppose P1 selects means F2 generation. Okay, here PA and PB are crossed, and we get F2 generation. So if P1 means F2 generation, so these are the plant 1 of F2 generation, plant 2 of F2 generation, plant 3 of F2 generation. This is how it is maintained. and they also name the row like this. Okay, suppose in the row it is like this p1 1 2. This is in the first f1 generation, in f2 generation, in a row plant 1 and 2 is maintained, then there could be p1 1 3, that is in a row plant 1 or 3 is maintained. So this is how they kind of maintain the record, generally by using unique identification on code number through the pedigree record book. Generally they mention all these things in pedigree record book. Okay, now what these records includes? These record includes first of all parentage. These records include parentage. Parentage means cross made and its f1 generation. Suppose this is p1 p2 and pa pb, and the f1 generation will be pab, for example. So this is the kind of the parentage, that is which parents have been used, through which this we have f1 f2 and successive generations have got. Secondly, we have to mention generations. Okay, we have to kind of give unique identification code to generation. For example, for f2 we have given p2, for f3 we have given p3, like this. Okay, so generation is from, especially from f2 generation onwards, it should be maintained very nicely. Okay, because we have to successively breed for generations, 3-4 generations, to achieve our results. So this needs to be maintained. Secondly, we also need to keep performance data. Performance data. This data would be of yield, disease resistance, plant type, all these things. And at the end we have to also mention, I'm rubbing it. You have to also kind of keep selection history. Selection history, that is which plant came from which progeny. Okay, there, because there will be various progenies, hundreds of plants you are experimenting. So we have to keep selection history, that which plant is coming from which progeny. Okay, so that suppose a plant is coming from B progeny, C plant is coming from D progeny. So likewise we have to maintain whichever number we want to give. Either we can give alphabets, either we can give number. That should be unique. So this is how kind of overall record is maintained in pedigree pedigree method. Okay, so I hope you understand this. If you mention all these things, you will get good marks. Now if you see the second part of the question, it is asked us to discuss the pedigree selection method. Okay, so we will discuss the procedure of pedigree selection method. Procedure. Okay, so the first step is to cross the parents. We have to cross the parents with desirable traits. Whatever traits we want in our progenies, we will take the parents and cross them into this approach. Okay, second, in f2 generation the f1 plants are selected. Second, we will in f2 generation, what we will do in f2 generation, the kind of there would be selection of plants. The f1 plants are self to produce f2 seeds. Here f1 plants are self to produce f2 seeds. Now what happened? There will be large f2 generation would be with us, and this f2 generation is allowed to segregation, and then we will select the individual plant. Individual plant on the best of phenotype. Suppose we are crossing for high yield, so we will see the plants with high yield. Okay, high yield or any kind good high disease resistance, anything. We will select the plant, and we will kind of select the plant on the basis of that phenotype. Selection would be on the basis of phenotype. Then step 3 would be in f3 generation. So what we will do in f3 generation? In f3 generation, each selected f2 plant, okay, is grown in separate rows. Whichever f2 plants we have selected will grow in separate rows. Okay, every inferior plant should be discarded. Suppose any kind of inferiority is there, it should be discarded, and best superior plants with best superior families are kind of allowed to grow. So this is the thing we will do in f3 generation. In next step is in f4 and f5 generation. So when it continues for f4 to f5 generation, so here we have to kind of what happens due to this further selection, these lines become more homozygous and uniform, and we have to just keep a pedigree record to kind of track the lineage. Okay, track the line of the ancestry. Then all these things needs to be generally here we have to track the lines, okay, because in F4 F5 generations we will get homozygous and uniform plants, so from which lineage they are coming, okay, we have to track them. Then from F6 to F8 generation, all the promising lineage, okay, in F5 and F4 F5 generation we will get some promising lineages. All the promising lineage or promising pure lines are yield tested and compared with standard varieties. They will be tested for about the quality. If the yield is our priority, it will be yield. If disease resistance is our priority, we will test for disease resistance. So all these things will be done, okay, they will be tested and they will be compared to standard varieties. Is suppose these are the standard varieties, here the yield is 10 kg, so it has increased or not, it is 10 kg, 12 kg, or it has decreased also. So what happens, we will do it. And in the sixth process, that is release of variety, if the tests are successful, if our varieties are successful, we will release the superior variety and we will do some multi-location trials, and then we will release it as a new variety. Multi-location trials are very important because it sometimes happens that in the field we are experimenting, it can give good yield, but in other locations it can the yield can regress. So all these things needs to be done. So this is the overall process of pedigree selection which is done. Now in the question you will see that we have to kind of make it a simple schematic diagram. So if you understand this process, it is very easy to replicate in a diagram. I will show you how. So this is how you make a diagram. First of all, we have crossed parents P1 F2, and in F1 generation we have laid the bulk F1 generation and kind of laid a lot of seeds to grow. Okay, so in F2 generation you will see that a lot of seeds we have selfed in F1 generation and produce a lot of seeds. With those seeds, a lot of plants are grown. Now, what are we doing in F3 generation? We are selecting the plants, okay, based on their phenotype. Okay, superior plants are selected. So you can see that plant, individual plant, and selection. So family grows in progeny, rows, and selection. What happens in F4? The family grows in progeny, rows, so that everything is well done. And in F5 you can see this, you can see the selection, how many plants were there, and through selection we have seen that very few plants, good yielding plants have remained. And then up in F6 generations we will do preliminary stage trials, select single plants, and then further yield trials, and in further generations we will do multi-location testing and we will release the variety. So this is the same process, but it has been written using this simple diagram. But just keep in mind, whenever you are moving from F2 to F12, we have to keep in mind in any method, okay, the plant numbers will decrease. Okay, if you keep just writing the process here and just keep decreasing those plant number, the diagram would be good. So I hope you understand how you can make a diagram. Now I think that would be enough for your answer. You have understood all the parts of the question. In conclusion, mention that yes, pedigree selection involves a systematic record keeping and stepwise selection, but it is very important to kind of breed good quality varieties. You can also give examples of some varieties. For example, Kalyan Sona is a variety of wheat. Sorry, not a weed. Kalyan Sona is a wheat variety which have been kind of breeded or which have been produced through this pedigree method only. So this is how you can approach the answer. So I hope you understand all the parts of the question. So that is all for today. If you like it, please press the like button. Please do share and please do subscribe. It takes a lot of hard work to make such videos for you on a regular basis. And if you like, if you share, if you subscribe, it motivates us to make more such videos for you regularly. So that is all for today. Have a nice day. Thank you.